Okay, if you could make your way to your chairs. Instructions for the speakers, don't wander too far over here because apparently the light is bad for the cameras, so we have to stand somewhere over here. Constrain your urges to move. Okay, so we had a question in the last session, what about applications? And this session is all about applications. Biodiversity genomics, meeting policy and practitioner needs. And it's a great pleasure to introduce Mara Lawniczak and Claire Carvell, who are going to talk about DNA barcoding, large-scale insect communities for biomonitoring. Mara.
Thanks, Pete. So as we heard so beautifully in the first session, DNA-based approaches are ramping up the pace at which we can discover and monitor biodiversity. And this is really exciting for science, but it's also sorely needed so that we can be better stewards of the natural world around us. In the UK, we are in a fortunate position at the Wellcome Sanger Institute to have made a major investment, a whole department focused on biodiversity genomics. And in collaboration with many other biodiversity organizations across the country, we're leading the Darwin Tree of Life project, which has come up already. And we're also leading a project called BioScan UK. This is inspired and aiming to contribute in some small way to the extraordinary vision and progress laid out by Paul earlier. And the way that it works, well, it's trying to measure arthropod diversity across space and time across the country. And in order to make this viable project, we put out an open call to try to reach organizations and institutes across the country who had a real need to measure arthropod biodiversity in their area locally, but didn't necessarily have DNA-based approaches accessible to them. So they essentially joined the project, and then now they volunteer to run a malaise trap for 24 hours a month, every month for the years to come. And they then take a catch, which is like this right here, a typical malaise trap catch for 24 hours, and without doing any identification, they just move the insects, each insect, over into a well of a 96-well plate. Then they send those plates to us at Sanger, and we image them automatically. So a photo is taken from above the well of every insect, so we have an image to go along with the arthropod. And then we leach out the DNA non-destructively so that the specimen remains intact, but we have a DNA aliquot for every specimen. We take a tiny bit of that DNA and we DNA barcode it. So this retains the links to the specimens, to the DNA, and to the DNA barcode. So we've been running so far for about 5 years. Our first 2 years, represented here, were pilot mode with just 10 partners trying to get us so that we were set up. And then we really launched the open call in 2023, and that's 2023's data, 2024's data, and we're now processing 2025 specimens. And you can see at the peak of summer in 2024, our partners were collecting 16,000 specimens, and these are all DNA barcoded now, and 16,000 specimens a month. So in total, our partners have collected over 370,000 specimens. We've DNA barcoded 200,000 of these. We've identified over 10,000 species or bins, as Paul was telling you about. And while only 22% of these are confidently linked to a single species in our UK species inventory, in a sense that kind of doesn't matter so much for monitoring change, because the barcode is enough to understand, have we seen this before? Is it changing its abundance or frequency over time and space? We also, because we're doing individuals, not metabarcoding, we can say things about abundance. So for example, 2,000 of our species have been observed more than 10 times each. 4,000 have only been observed once, and we've identified 1,000 new bins, added 1,000 new bins to the IBEL bin count. Last week, through BGE sponsoring, we ran a taxonomy-thon on our specimens where experts in Diptera and taxonomic experts in Diptera and Hymenoptera came to the Natural History Museum in London and looked at several hundred specimens and managed to put taxonomic names, Linnaean names, on 188 species with bins, barcodes, and that affected 8,000 specimens in our collection alone. So we are firmly in the era of DNA-based biomonitoring at unprecedented scale with affordable costs, and this is where we're starting now with BioScan UK in a small way in the UK. We have a long way to go before we can really get that spatial temporal monitoring going. But there are lots of historic projects that are DNA enabled, and now I'm going to hand over to Claire to tell you about how we've used this BioScan infrastructure with the DNA barcoding at scale to investigate the bycatch from the pollinator monitoring scheme.
Thank you, Mara. Thank you. So, hello everyone. So the UK pollinator monitoring scheme is aiming to effectively establish how insect pollinator populations are changing across the UK. It's been established since 2017, and it's co-funded by the UK government through the Joint Nature Conservation Committee and the UK Centre for Ecology and Hydrology, which is the institute where I'm based. All of the data from the scheme is published under open government licence, and we're working with our government to develop official statistics and indicators of pollinator status and trends from the data that we generate. So POMS includes two large-scale citizen science surveys, which collect this systematic data on annual abundance of bees and hoverflies as the target species, but also a whole host of other flower-visiting insects. The main survey is what we call the 1 kilometre square survey, and this is set up over a stratified network of 95 sites across the UK. So you can see the map on the right here. These sites are stratified by country area and the relative cover of semi-natural and agricultural habitats in the UK. So we want to look at this kind of national level surveillance to understand trends over time that are representative of our landscape. And the surveys are repeated 4 times a year from May through to September. This involves setting out these coloured pan traps at 5 locations across each one of our 1 kilometre square sites. They're left for a 6 hours, so a sort of fairly straightforward 1-day exposure period with water to trap the insects, after which those sampled insects are transferred into 70% ethanol. And they're brought back to our centralised lab. So most of the actual surveys are conducted by volunteers who adopt a square near them, and any of the gaps are then filled by surveyors on our team. So between 2017 and 2024, we've generated data from more than 1,723 surveys. We have in excess of 8,500 archived samples, one sample representing one of this triplet of pan traps you can see, and that comes to around 30,000 bee and hoverfly specimens from literally all corners of the UK. So our current sample identification pipeline is focused on morphological identification of key pollinator groups. And it effectively involves sorting through each of the samples that are represented by the tubes. You can see here, single tube, a single sample, and counting out every insect according to its broad insect group. So this pie chart shows the sort of typical makeup of one of these pan trap samples by these broad groups. But only the target pollinator groups, the bees and the wasps shown near the top, which are kind of making up about 4% of each of the samples here. So this is really a very small proportion of the data that we're collecting, and in order to maximise its value, we really want to try and understand more about this kind of vast array of other species, particularly the non-syrphid flies, the Diptera, other Hymenoptera, the beetles, and so on. And so there's this kind of huge amount of unknown diversity and ecological complexity among this bycatch, which is, again, has the potential to be explored and understood by DNA barcoding approaches. So this is where we collaborated with Mara and her team to run a small pilot study linking up with the BioScan project. So we first tested, we wanted to test whether the DNA barcoding pipeline developed by BioScan could actually be used to identify POMS pan trap specimens, with the overall aim of really wanting to understand if DNA-based measures could tell us about changes in insect community assemblages over space and time. So for this pilot, we took 155 samples from 31 survey visits across the 4 sites you can see here. So the data I'm going to show you very briefly is from these 4 sites shown in yellow, from the north of Scotland to the south and central England. And that was involving plating, individually plating 10,000 insects using the protocol that Mara's just described. We're pleased to find a really high sequencing success rate of 93%, which kind of confirms that the quality of DNA within these archived samples that date back to 2017 is compatible with that BioScan protocol. So that is a sort of positive starting point. The results are quite astounding in many ways. We found overall from just those 4 sites, we've identified 135 insect families represented and sequenced from within those samples. And you can see some examples of some of the 25 most common families here. The reference databases were usually accurate to family and almost always accurate to genus level, but it's definitely not always accurate to species level, and this is something we're investigating further. But breaking down the data according to this sort of relative abundance here of the different insect families across the sites and across time, you can see this is purely based on the DNA sequence data at family level. And they show you've got some really interesting differences and contrasts already between sites, between, for example, the two more semi-natural sites in the north that are more dominated by some of the Anthemidae and the Phoridae, and then the agricultural sites represented in the south, more dominated, for example, by the muskid flies. But this next plot really starts to show us where the DNA approach gives us insights that we simply wouldn't have with the morphological approaches on their own. So this is a sort of effectively just unique number of DNA sequences or DNA richness, sequence richness per sample across time, the 3 years represented across the top and across the 3 sites. And in total, there were more than 3,500 unique sequences of insects emerging, again, from just these 4 sites alone in just 3 years' worth of data. And we're able to detect some significant changes over time in 2 of the sites and significant decline in insect DNA richness, for example, at the Goring Heath site there in 2022. And then finally, another snapshot of the data from this pilot study. This is an example of looking for metrics representing a whole community composition and how this varied within sites, and in particular within the season. So the coloured ellipses here are showing you contrast, especially again at these 2 top sites between the seasons, showing how important it is to sample these ecosystems and these communities across time through a season, because insects, of course, are changing and moving all the time in response to their environment. And again, this sort of community diversity metric is genuinely only achievable by the barcoding and through the bioscan pipeline in this particular case, and it wouldn't be available even from meta barcoding alone, because we're using the abundance of the associated insects within that sample. So I think, yeah, just to conclude, really our conclusions mirror many of the points that have already been made this morning, that DNA-based species discovery and biomonitoring can indeed transform these huge challenges that we have, particularly in the insect world with taxonomic uncertainty, from being a kind of barrier into a really sort of structured pathway for improved biodiversity intelligence. And this is especially possible given these large-scale citizen-led sampling efforts and carefully curated archives. We again sort of emphasise that we need continued investment in reference libraries. That was definitely borne out by the data we had here. It's still needed, and it must include continued collaboration with taxonomists. We're showing that metrics describing DNA species richness or diversity of these terrestrial insects are revealing differences in insect community assemblages over time and space. But really we want to move on now and think more sort of creatively about the whole kind of the question around ecosystem function. Can we extend this to increase our understanding of resilience of ecosystem functions? We have enough an ecological understanding to associate ecosystem function to many of these species groups. And finally, of course, how can we scale up these DNA approaches and operationalise them within our national scale insect monitoring schemes and deliver policy relevant indicators and inform our emerging biodiversity targets. I just finish by thanking all of our collaborators, co-authors, funders, the taxonomists we've worked with, and especially the volunteers who've been collecting the data. Thank you.
And a perfect example of keeping to time. Any questions? Time for one quick question? Maybe I could just ask a quick one. I was kind of fascinated by the discovery of new bins. Do you yet have a kind of sense of the scale of hidden diversity in flying insects?
Well, we've had a little bit in the UK, but nothing like what Paul has shown. Yeah, I think of the 1,000 or so bins that we've seen where it's new, they're adding new bins to the records. About 2/3 of them have only been are specimens that have only been observed once, but a third are more common.
Any more questions? In which case, we're running slightly behind, so many thanks to Claire and Mara, and Jose will take that microphone. And next up is Leif, who's going to speak about how population genomics can promote sustainable use of natural populations.
Okay, Leif Andersson, Uppsala University. So fisheries has been extremely important for food security for thousands of years. So this illustrates herring fishery in Sweden in the 16th century. But unfortunately, we have a very sad track record of overexploiting fish populations. Maybe the most famous one is the cod populations in Newfoundland, where there was this enormous catches of cod, particularly here. So I was a kid in the 60s, and there was these fish pins that was swimming over all the world, but led to a total collapse of the population in 92, and it hasn't returned back since then. So what is really important with regard to fisheries is that you set fishing quota. And to do a fishing quota, ideally you would like to do it on the biological unit, this subpopulation, because many of fish populations are subdivided into different populations. So sustainable exploitation of natural population requires detailed knowledge of the population structure. So is there one population or are there many? And the way to do this is to calculate allele frequencies at polymorphic loci, because if there's a single population, you don't find any differentiation between different catches in different areas. But as soon as you see difference in allele frequencies, then you know that these are distinct populations. And DNA-based genotyping is the most cost-effective method to generate such data. And the challenge here is that marine fish that are heavily exploited are, of course, abundant. I mean, that is the reason why we go after them, because they are a huge biomass. And they're often migratory, and that leads to gene flow between populations and very low genetic drift, which means that allele frequency at neutral loci is often very similar, even between distinct populations. So low genetic drift and gene flow restrict genetic differentiation. And a problem we have is that we lack reference genomes for many of the marine organisms that were exploited by humans. So I would argue that for these species, since it can be difficult to find the most informative markers, it's very valuable to have high-quality reference genomes. And I will give some examples of that add to the possibilities. However, I would say that reference genomes are useless for protecting biodiversity in marine organisms unless you combine it with population studies. So the reference genome is a required tool, but it's not sufficient. I think that is important to keep in mind. So we published last year a roadmap for how we think this should be done. And you start with the genome assembly. In the next step, you do this population study. You sample fish or crabs or whatever, shrimps, what you're interested in. And then and preferably be on the spawning ground, so you know that you are catching a spawning population. And then you do whole genome sequencing, aligning the reads to the reference genome. And then you could compare populations and identify the regions of the genome that distinguish different subpopulations. In the third step, because in practice, if you're going to apply this in commercial fisheries, you can't do whole genome sequencing. But what you do then is to select the most informative genetic markers and use a SNP array to test those. And then you evaluate those on different populations. And in the fourth step, this is then applied to fishery management. And during a couple of years ago, we were involved in designing a multi-fish SNP chip, which contains thousands of SNPs from 7 different marine fish that is exploited in commercial fisheries to get the cost-effective way to use SNP chips. But the only species that this has been fully utilized so far is the Atlantic herring, and I will use some examples from that. So this is data from Atlantic herring. So we have been doing whole genome sequencing. So this phase where we have sampled herring from all areas. So this is the Baltic Sea, which is brackish, North Sea, Norwegian Sea, around UK and Ireland, Iceland, Greenland, but also on the American side. And basically what you see here is a principal component analysis where we have selected about 800 of the most informative genetic markers to look at the population structure. And what I would like to say then is that herring is one of the most abundant vertebrates on Earth. But what you see here is the population structure, and in fact, herring doesn't have much differentiation at neutral loci. So what you see here is in fact reflect genetic variants that is involved in ecological adaptation related to climate, salinity, spawning conditions, light conditions that reflect this. And this can now be monitored using this SNP chip that I mentioned. So if you take the North Sea, which is a place where the fishing fleet from this area is catching fish, and these different populations that you see, these are the Irish and British populations, and this is from transition zone, this area. They migrate out and feed on the plankton in the North Sea. So when you're fishing here, you're mixing all these stocks basically. And this is now the most cost-effective tool to distinguish a different population. So in principle, what you can do now is to set the quotas based on the biological unit rather than to the geographic area where you are fishing. So one reason why high-quality reference genomes is very valuable is that they allow the detection of large structural variants like inversions. So what you see here is 4 inversions that is really important for climate adaptation in the Atlantic herring. And you could only see that when you have a linear organization of the genome. So this is chromosome 6, 12, 17, and 23. And what happens with an inversion? So inversion is when a piece of the chromosome has flipped orientation. And what happens then is that gene variants within are inherited as a block. And that is what you see here, that all the SNPs in this block reflect these different configurations. And we couldn't see that until we had a chromosome level assembly. And these are really important. I could also say that these inversions have evolved over the last 1,000,000 years. So they're 1,000,000 years old, and they have contributed to climate adaptation in this species for the last 1,000,000 years. Here I would like to illustrate how we apply this SNP chip. Sorry for this resolution, it's not super, but so we have used this SNP chip to scan the population spawning in the Baltic Sea. So what we have here is that we have spring spawning Baltic herring and autumn spawning Baltic herring, and they've been genotyped for 4,600 SNPs using this SNP chip. So autumn spawning and spring spawning is 2 different reproductive strategies where the fish is utilizing different plankton peaks. And what you can see is that the autumn spawning is much more rare than the spring spawning. But 100 years ago, it was the other way around. Then the autumn spawners were dominating because the ecological conditions were more favorable for this group. So what happens in the next 100 years, we have no idea. But this is really important biodiversity in this population. So then what we can do with this, so remember now, most of these SNPs are in fact in regions under strong natural selection. So when we compare spring spawning and autumn spawning Baltic herring, we get all these peaks, which has to do either with controlling spawning or controlling fitness, depending on when you are spawning, because different populations utilize different plankton peaks. And you could see the significance values here are quite impressive, 10 to the minus 1,000 for this. And that is because at this peak loci, there are complete fixation for different alleles. But what I would like to say here is these are currently managed at one single population, although they're clearly 2 distinct populations. So I think what this illustrates is the important biodiversity that exists within species. And the UCN Red List totally ignores the importance of intraspecies biodiversity. So what you see here is this, So herring is classified as least concern. But today there is a meeting in Stockholm discussing the decreasing populations of marine fish in the Baltic Sea. So I think this is a very important aspect to take into account. Something which is also important to notice is that the population structure varies considerably between species. So what the procedure I have described to you illustrate how you could explore this. So Atlantic herring has an amazing diversity of subpopulations. Then we have European sprat, Atlantic horse mackerel, and Atlantic mackerel, which is in fact one of the case studies in the BGI project. And these species show structure, but not at all at the same level as these species do. And the most extreme example is the European eel, which is one panmictic population. All are spawning in the Sargasso Sea. And then, so there is no genetic adaptation to the big diversity environment that they are exposed to. So, but this is also really important information, because that means that all countries that is managing eels need to work together, because we have one single population in Europe. So when glass eels are served as tapas in Spain, it will affect the recruitment of eels the next generation in Scandinavia, for instance. So what I think is the reason for this variation has to do with the difference in reproductive strategies. So my final strong opinion here is that generation of high quality assemblies combined with population studies should be a top priority for all economically important species to provide the tools for sustainable exploitation. Then the next challenge is, of course, to convince politicians that this is important, to follow this. We know about this structure in the Baltic, but this has not yet been implicated in the management. Thank you very much.
Any questions? Paul?
So that's fabulous, Leif. Question for you. I wouldn't want to do it on herring, I don't think, but those SNP arrays, potentially you could use this in a sort of a genetic mark release recapture to estimate population sizes, could you not? I mean, if they were really valuable fish with, I'm thinking tuna or something like that in the Mediterranean, to estimate population size, not just differentiation of stocks, but the number of individuals.
Yeah, based on what do you mean, how should you do it?
Well, I mean, I think the way they're doing it in Australia is with large capture fishery that goes and gets a certain number of young fish.
I mean, the classical way is to tag them and release and then see.
Yeah, the problem with tags is people pull the tags off when they're trying to underreport. So I think, you know, there's a big project on
herring is a really
tunas.
difficult species to work with because it's so sensitive, so it's difficult to tag. So when you take up herring, they tend to die. Because herring is a big schooling fish. So they don't have much armor in the skin, like a cod or a salmon. So they are difficult to do in that way, in fact. So, but the population size of herring is enormous. We think the census population size is about 10,
I wouldn't want to try herring.
10 to the 12.
Wouldn't want to try herring, but I'm thinking about the tunas in the Mediterranean.
Yeah, absolutely. I mean, in other species, you can definitely do that type of thing. Yeah.
Yeah, thank you.
Any more questions? Camilla? We've chosen somebody furthest from all microphones.
Thanks a lot, Leif. This is a perfect example for when we talk about genomes and genomics. I completely agree with you. To make genomes really flourish and have a purpose, we need to have the population representation there. What you have done is what I call the genome enablement of a species, where you try to represent the entire genomic diversity across the species. But also, I mean, in your graph, if you would put the amount of effort that you needed in each step, I suppose this one was a giant one, right? Getting samples from all populations. Sequencing, I mean, as you sequence populations, especially at a higher coverage, I don't know if that was the case, that's probably also was very costly. And that's in the That's when the reference genome becomes a tiny part of a very big project. Very important, right? Because that's the anchor, but it's costly in terms of costs. It's actually, I suppose, a tiny part. I actually wanted to ask you to comment about this because, I mean, people think everybody agrees it's important, but many people fail in do a proper representation in populations before going, for example, for a SNP chip or something that is more rapid and less costly, and in many cases could just lead to some biases, for example. What is your take in this process, and have you done before some other order of this process, and do you see the difference now that you have the entire process of genome enablement from beginning to the end?
So you should remember that monitoring herring populations and other fish populations have been going for 100 years because they are so important to our economy. So the commercial fisheries are sampling, and they measure the age, and they try to figure out which population the fish is coming from, and then to use that to set quota. So they are monitoring these populations, and they've done it on morphological measurements and so on. So what we did to do this really extensive screening was, so we didn't sequence many whole genomes individually. So we took the population and isolated DNA and pooled the DNA, and then we sequenced each pool to 30x coverage, because then you read 30 reads from at each locus on average, and that gives a really nice resolution of the broad picture. So I think I think you can do that in a very cost-effective way to do these population scans, because if you work with the fisheries, you can get the samples easily. Then you could do this pooled whole genome sequencing, and in the next phase you could do SNP chip analysis, which is become quite not so expensive. So compare with, I mean, they already today invest in doing this, and for the herring story, this is more cost-effective now than the procedure they have done in the past. So I think there is a great potential for this. So.
Thanks.
Thank you very much. I'm afraid we're going to have to move on. So thanks to Leif. And Christina is going to talk about genetic diversity indicators.
Yes, and I just need to figure out how to do this. Yeah, there you go. And thank you, Leif, for setting the scene. It was a perfect introduction to my talk today. So I'm going to talk about genetic diversity, indicators for genetic diversity, what is currently ongoing in Europe, and also how it links to what's going on a global scale. So there are 3 pillars of biodiversity. There is ecosystem diversity, species diversity, and there is genetic diversity. And as we just heard, genetic diversity is super important, as it underpins the long-term survival, adaptation, and resilience of species and populations, and also entire ecosystems. Unfortunately, here in Europe, we see more and more degraded ecosystems. But fortunately, as we also heard earlier today, there is the new EU Nature Restoration Regulation, which, together with genetic diversity and a focus on that, will allow us to restore and support healthy ecosystems. One key example of that is the devil's bit scabious from Flanders here in Belgium, where genetic diversity is directly linked to healthy ecosystem. So, unfortunately, we are also losing genetic diversity. We published a paper earlier this year where we looked at more than 600 species in a large meta-analysis. These species were looked at over at least 2 time points, and we found a pervasive loss of genetic diversity. But there is also hope. So fortunately, conservation actions can mitigate the losses of genetic diversity. So we need to take action, because otherwise we will continue to lose genetic diversity. We also published a paper last year where we actually looked at genetic diversity and the DNA-based monitoring going on in Europe. Unfortunately, not much is going on. When I say monitoring, I mean over at least 2 time points, so on a temporal scale. But there were a lot of kind of single time point studies, so which can serve as kind of the first baseline for temporal monitoring. So that is one good aspect to take from this study. Genetic diversity is really important, and it is one of the 3 pillars of biodiversity, also recognized by the United Nations Convention on Biological Diversity. So the Convention on Biological Diversity is a kind of multi-nation treaty where 196 nations have signed this treaty. So for the first time ever, and the CBD dates back to the early 90s, for the first time ever in 2022, wild species and their genetic diversity actually made their appearance in the global biodiversity framework of the CBD. But without indicators and monitoring, we cannot really follow the processes that threaten genetic diversity, such as loss of genetically distinct populations, reduced population size, climate change effects, and alien populations. So for the CBD and the Global Biodiversity Framework, 3 pragmatic indicators were proposed. Indicator 1 is effective population size 500. Indicator 2 is the proportion of populations maintained. And indicator 3 was monitoring based on DNA-based techniques. So indicator 1 and 2 can be assessed both with and without DNA-based data. 2 indicators made the cut. And indicator 1 is now the headline indicator for the CBD. And indicator 2 is what we call a complementary indicator. So I want to show this slide because it is fantastic that we now have indicators in the CBD and that they can actually be used both with DNA-based and non-DNA-based data, so proxy data, which allows them to be globally usable. So they are affordable, they can really be scaled to hundreds and thousands of species, but they are kind of a quick multi-species diagnostic of the genetic health of populations and species. So DNA-based studies are a complementary method. The indicators are not a substitute for DNA-based studies. They are complementary and they offer different things. So it's important for me to stress that DNA-based studies offers a more kind of finer, detailed picture of what is going on in populations and species. So in terms of inbreeding, load, introgression, and also genetic monitoring. And most importantly, it can directly inform conservation actions. So it's super important that both of them kind of are used in parallel. So now you have a bit of a background on genetic diversity and indicators, and I'm going to showcase a couple of the efforts going on in Europe and also link it to what's on a more global scale. So there are a couple of European funded projects. One of them is the Biodiversity Plus project, Genetic Indicators for Nature Monitoring. It's a large transnational research project with more than 11 partner institutions in 8 countries, and we are more than 30 people now. And we are a very diverse team with a lot of geneticists, but also social scientists and other backgrounds as well. And it's directly based on the need for countries to report the genetic indicators to the CBD, where the first time is next year and then every 3 years. GINAMO's mission is to co-create user-friendly workflows with country stakeholders, enabling them to report on the CBD genetic diversity indicators and also use them for monitoring. And I'll come back to that in a second. So what we do in GINAMO is that we determine the best practices to obtain accurate and robust effective population sizes, so indicator 1 would reference DNA-based data. We also do the same with proxy data, and also look at the indicator 2, the proportions of populations maintained. And we use best practice recommendations feeding in from stakeholders as well to develop standardized and user-friendly essential biodiversity variable workflows, and also develop pipelines that are interoperable and all, of course, in agreement with fair and open data. So the crux of GINAMO is to work together with countries. So we work with 5 countries where we have a lot of different stakeholders, from nature managers, NGOs, to people working on red listing, the CBD national focal points, even politicians involved in our stakeholder groups. And where we do an inclusive, facilitated workshop process with them to identify what are their resources in the given countries, what are their needs, what are their challenges, and which challenges can we as GINAMO help with in their kind of path towards using indicators and genetic diversity for monitoring. And then once we know that, we then work with the country stakeholders to actually develop something that is useful for them, so it fits their needs and resources, meaning that it's much more likely that what we end up producing actually fits their needs and will be used in the future. So these are some of the workshops that we held in Sweden, in France, in Belgium, in Italy, and in Norway. But GINAMO is just one project. Unfortunately, and you've seen this logo before, there are other great efforts ongoing as well. GENOA is an EU cost action network which runs in parallel with GINAMO. And with GENOA, we're many more people and many more countries involved. So we're able to kind of ramp up and scale up our efforts to help countries actually use genetic diversity for species assessments and protecting biodiversity. So now we switch from global treaties to a global union, so the International Union for the Conservation of Nature, IUCN. Most of you might know the IUCN Red List for assessments of species. And a recent study had a look at the Red List assessments and whether it rightfully correlates with genetic diversity loss. And the paper showed that many species that are listed in least concern or near threatened actually have low genetic diversity and the low genetic diversity indicator value. So the same goes for, so the red list of species does not take genetic diversity into account, and neither do the red list of ecosystems. But the IUCN Conservation Genetics Specialist Group proposed a motion to the upcoming IUCN World Conservation Congress next week on a task force that is set to develop a global standard for genetic diversity assessments, so that we can now also hopefully have genetic diversity assessments for species and their populations. And the voting on these motions ended last week, and there was fantastic support for this motion, so now we'll proceed internally in IUCN and hopefully become a resolution. I also want to just showcase 2 resources as well, and then I'll come to my, I'm good on time? Good. So IUCN has a wealth of resources. Some of them also have a link to genetic and genomics as well. Back in 2022, we published guidelines on selecting species and populations for monitoring of genetic diversity. And in there are some great examples of the use of genetics and genomics for monitoring biodiversity, for instance, in Switzerland and in Sweden. So go have a look. They're freely available on the IUCN library portal. And hot off the press, it will come out tomorrow, the new IUCN guidelines on biobanking for conservation purposes, because without samples we can't do any genetics. So we have compiled guidelines that kind of compiles all the information that is out there on best practices, procedures, legal aspects, regulations, etc., and also outlines kind of a roadmap for a global network of biobanks. So there are some good resources in there as well. So in summary, hopefully you will go back home today or to your hotel or wherever you will go and thinking genetic diversity is important. Unfortunately, we do have indicators, and there's also genetics and genomics DNA-based studies being done to monitor populations and species. And there are a lot of active communities out there in Europe and also globally that work with countries on reporting and using genetic indicators for the CBD. And again, there are active communities and networks that focus on the scientific work behind to improve the indicators. And there is definitely also a global leadership in practices and standards developing tools to ease the application of the indicators. There are a lot of resources. We've also heard a lot of resources today, but I would still argue that there is a huge need for compiling and making all of those resources findable. And also, countries are diverse, their needs and resources differ. So there is a huge responsibility from the countries that do have the resources, the know-how, etc., to transfer that knowledge to other countries. And then, of course, there's the issue of finances as well. So that was my key takeaways, and thank you for listening.
Thank you, Christina. Got time for one quick question and one quick answer. Andy caught my eye first.
Thank you. I'm Andy Nisbet, work for Natural England in UK, where I can run several large monitoring programs. So this is brilliant. It's really great to see kind of indicators, but what do you think a good national monitoring program to generate the data to service those indicators would look like?
It doesn't really exist.
Imagine one.
But ideally, I would say one with a long-term funding, because I've seen over the years a lot of good initiatives popping up, but all of them as pilots. So they simply run out of funding to do anything. So I would say funding is key. And then there are larger scale, like in Switzerland, where they use full genomes and the full Monte. And then there are other initiatives elsewhere where the costs are lower, but they do provide an insight into the state of genetic diversity of species and populations.
Thank you, and I'm afraid we have to move on now, so a round of applause. And Christine's going to talk about monitoring alien species, and you can pass the microphone directly.
Okay, so I'm new to BGE. I'm a botanist, curator of the Vascular Plant Herbarium in Trondheim, and a lot of my work is focused on alien species in Norway and in the Arctic. And thank you very much for inviting me to come here and present some of our work on alien species and how the use of DNA-based methods is an essential part of our efforts. So I represent a team of researchers, and our goal is to inform management strategies to mitigate the impacts of invasive alien species. So everyone knows that the number of invasive species and their impacts are increasing. Managing them requires international collaboration across people, scientists, stakeholders, and policymakers. And they are specifically targeted in the Kunming-Montreal framework. According to Target 6, we shall reduce the introduction and the establishment of invasive alien species by at least 50% and minimize their impacts by 2030. That's 5 years from now. And we have the IPBES report on invasive alien species providing evidence, tools and options to help governments achieve these very ambitious global goals. And in Norway, our contribution to Target 6 is to reduce, of course, their impact on biodiversity, and also to reduce the rate at which such alien species are introduced and become established. So this is a classical invasion curve showing an alien population response over time from introduction to invasion. And it also shows the cumulative costs of management inactions. So our work is mainly focused on the pre and early introduction phases, because we need to know which alien species we are dealing with. We need knowledge about active pathways of alien species. We need to detect them, we need to identify them, we need to risk assess them and their pathways as early as possible. And only by knowing this can we do something cost effectively to reduce their impact. So one very well-known pathway for both intentional and unintentional introductions is through the imports of horticultural plants that are rooted in soil. And this is big business, it's increasing. You can see on the left here the amount of horticultural plants with soil that has been imported to Norway since 1988. And you can also see the value of this import. So we were tasked by the Norwegian Environment Agency to first develop and then run a program to monitor this important pathway. And our goal is still to get systematic knowledge on which species are introduced, which exporting countries do they come from, are there variations over time, and which of these species have the potential to establish in Norwegian nature and cause harm. So we collaborate with one of the largest importers of plants in Norway. We take our samples immediately upon arrival, before the plants are repacked and transported all over Norway to be sold. And the top 5 exporting countries to Norway are the Netherlands, from which we import 2 to 3 times as much as we do from Germany, Denmark, Sweden and Italy together. And while all soils imported to Norway must be of European origin, there is no information about their source of origin, nor any possible biosecurity treatments of the soil that follows the imported plants. And this brings us to our first challenge, because from these imported soils we need to extract and identify all viable seeds and as many live invertebrates as possible, so that we can estimate introduction rates. And based on our monitoring data from 23, Norway imported at least 800 million viable seeds and more than 1.9 billion live invertebrates as contaminants only through this pathway. So I have some graphs here that summarize our results for 10 years of monitoring. We see little or no flattening of the cumulative numbers of introduced species. What we also see is that we're not only finding alien species of European origin, the whole world is the source. And we don't have too many taxonomic specialists in Norway. It also takes a lot of time to morphologically identify all of these species from all over the world. So we have been implementing DNA-based methods to both to broaden and to speed up species identification. So this is an example of our DNA barcoding of seedlings, because we germinate seeds from the collected soil samples. We try to identify the seedlings to species as soon as possible, but this can take 24 weeks or more for difficult cases. And by using just Sanger sequencing of very common markers, we have compared our sequencing success rate of these markers, shown in purple here in these circles, and also how well their taxonomic resolution matches our morphological identifications. So for example, for MAT-K, we see that the results agreed with the morphological species identification in 53% of the cases, and for RBCL, we only got a match in 44% of the cases. And for some seedlings, the species identification differ between genetic and morphology. No surprise. We grouped our sequences here per morphospecies. We have green colors indicating where one marker gave the same identification as the morphological identification, and then purple indicates that we have different species identified by different methods. Of course, we see reasons for the lack of correspondence here that has already also been mentioned here. We have the non-specific amplification of non-target organisms. That's no surprise that we sequence fungi instead of the plant. We also see that the marker cannot distinguish between closely related species. We only get to the genus. And then we also have found that we have incorrect morphological species identification, which of course can happen when two species are relatively closely related and share many morphological characters at the seedling stage. But the last lack of correspondence reason here, we have not found any cases of misidentified reference sequences, so that's good. And then we have another challenge, is that we want to identify extracted live invertebrates across all life stages. And this is, in many cases, impossible to do based on morphology, but by DNA metabarcoding of all extracted invertebrates from the soil samples, we can detect many more species than we have the capacity or ability to do based on morphology alone. So we're scaling up the amount of biodiversity that we can identify to species, but we're also gaining a much broader knowledge about their haplotypes. And this allows us to start investigating cryptic invasions, because we see alien haplotypes of species that are native to Norway being imported. And we don't know much about the extent of this genetic pollution, but it can potentially have a very large impact on threatened species, for example. So we need to continue to build and curate the reference databases. And in our case, since we find species from all over the world, we are still only able to identify a subset of our taxa to species. And that a challenge for a program like ours is that we need reliable estimates of introduction rates and also trends for alien species, and DNA metabarcoding can only provide a semi-quantitative estimate of individual numbers. And the last challenge I would like to mention is our need for early identification of potential high-risk species and their sources. And we need to do this if we are to do something cost-effectively to reduce the impact of invasive species. And such a horizon scanning is a crucial tool for preventative management, and it allows us to draw up prioritized lists of alien species to follow. And by DNA metabarcoding the imported soils, we can identify and characterize the species composition at the site of origin of the imported plants and the soil. And of course, we find a lot of environmental DNA that comes from fragments of species that we never extract live. But this DNA also represents hard to observe life forms like fungi or rare species that we by chance miss extracting alive. So we recommend our environment agency to keep prioritizing the use of DNA metabarcoding of soils so that we can build up the knowledge about this larger source species pool of potential invasive alien species that may be introduced through this very active and important pathway. So we have several monitoring programs running in Norway. One that focuses on the introduction of alien species, which I just presented. And then we have another one focusing on the early detection of established alien species. And then we have 2 programs where there's a general monitoring of the Norwegian nature. And that can also capture the dispersal of alien species into the nature. And since we're now using DNA metabarcoding across these monitoring programs, we can start detecting the cryptic invasion of alien haplotypes. And I just included this Venn diagram as a little example of what we have, how we have looked into this, because we compared the haplotypes that we have found in a subset of our soil samples with all barcodes from all the other monitoring programs of Norwegian nature. And we were able to identify close to 1,000 unique haplotypes from 110 species that are native to Norway, but may represent a cryptic invasion. So this was a brief introduction of more than a decade of our work. Our key priorities moving forward include more use of DNA-based methods in parallel with traditional species identification. And what we see is that we need to make a full use of all synergies between our national and also international monitoring programs, and also with European and international research infrastructure and networks, if we are to deliver anything for target 6 before 2030 or even later.
Fantastic, thank you. Time for one quick question. Brad?
Thanks. I'm Brad Schaefer from University of California at Los Angeles, and it was a great talk. So, presumably, I'm curious about the movement of haplotypes of native species, but non-native haplotypes into your system, into Norway. And I mean, under climate change, we would expect a lot of that, right? We would expect new haplotypes to come in. We might even want that to happen if they're being selected for. And so are there, I guess, 2 questions. One is, do you see any evidence of previously non-native haplotypes sweeping across, or are they just individual? Or do you have the data to say that? And second is, do you view that as a bad thing?
Well, we haven't found any evidence because we have just started to look into this. We're not sure if it's positive or negative, but what we see is that we find red-listed species with a high category being introduced to Norway, and that might not necessarily be a benefit for the Norwegian populations.
So I'm afraid we're going to have to move on now, but we'll have time at the end to come back if there are further questions. So thank you very much. And now Alice is going to give us the perspective from business. The green one.
Okay, oops. Okay. So I'm Alice Valentini. I come from a private company that is called SpyGen. It's a French company specialized on the use of environmental DNA for biodiversity monitoring. So just a brief definition about what is environmental DNA. It's the genetic material that can be collected directly from the environment, such as water, soil, or air, without capturing or isolate the organism. It's a mix of living cells, for example, bacteria, or trace left behind organism or shed material, such skin or mucus coming from fish. And it's a mixture of DNA from many different species. So the revolution of environmental DNA starts more or less in 2008 with this article of Fisetola about the using water sample for detecting invasive species. I think all my talk is about microorganisms, because microorganism environmental DNA was using since 1870s, I think. So in 2008 was the first paper talking about environmental DNA and its use for a conservation purpose. The definition of environmental DNA came up in 2020, but the revolution started on nearly 2020 with a new journal only dedicated to environmental DNA. And the articles are increasing every year exponentially. At the same time, different groups are built up to work on environmental DNA, where the researchers on environmental DNA group together and discuss all about the optimization of the method, what we know, what we don't know, and they are still growing and different programs are going on. At the same time, there is also the business that come out with the environmental DNA environment. We were the first company that come out, but this is not by chance, because both founder, me and Tony DeJohn, we were former PhD student of Berta Berlet that did the definition of environmental DNA. After several groups come out, I divided by early entrants, mid-stage player, late entrants, but and there is not an exhaustive example. There are just few groups that just the company that pop out in my mind when I did this presentation. And so I divide by early entrants that come out in just after the definition of environmental DNA, mid-stage player, a late entrant, and the late entrant are not people that are new to the environmental DNA environment, because the CEO of Simplex DNA, it was Christy Diner, that is one of the pioneers on these topics. And at the same time that new groups are come out in the market, also well-established market players like SGS Aerofunds started to propose this service to different groups. And this was because there was a need, because in the university people have their genetic lab, but it was not the case for all the players in the biodiversity monitoring. And that's why the need all these companies are come up in the market. By why? And we start in 2011 with one company, and now I think we are more than 40. So this is because eDNA is very relatively easy to use in the field. There are different protocols, but some of them are so easy that even a child then can use it. And it's very useful to detect hard to identify species. For example, in this case, species that are very difficult to identify taxonomically, and they can be very small and completely dispersed in the river. And since the use of environmental DNA analysis in France, there is a significant increase on the collection, the collected French National Inventory spheric data on this database. Can help to do large-scale standardized survey. For example, in these two papers where it was used for mapping the coast-to-coast of Canada and to monitor all Japan. And this is another example that we did in collaboration with the University of Montpellier, the group of Marbec. We are Mediterranean and Andromed, Vigilife, and Open Ocean and the CEF, and eDNA was collected all around the French coast in different type of environment, coastal, port, and lagoon. There were a campaign of over 600 eDNA samples, and was done in less than 3 months. And it was possible to describe the species detection of different groups for fish, and it was possible to identify the hotspot of biodiversity and also the place where UCN species were present, and to have a better understanding of conservation purpose. It was recognized to be better tools for species richness because have a greater species richness and detection probability, and at the same time is cost less and for less effort in the field. But this is not true for all the species, because for fish this is the case, but for example for bird, traditional monitoring is the best. However, we're talking about environmental DNA, so it's a tool, but you need to know how to use it, because behind the name environmental DNA there are several protocols and several equipment, infrastructure, and you need to really fine-tuning what you are looking for. Your scientific question will determine the choice or all the things that you will do for the analysis in the lab, the protocols used in the field, and different things. For example, just focusing on the volume of water to be collected, in the practical guide of DNA-based method for biodiversity assessment, it was wrote that you can collect between 50 milliliters to over 100 liters. This means that it's a huge difference on the strategy that you will use, but this choice will depend on the sampling strategy, if you want to do an integrative sample or just have a different point sampling. The practical constraint in the field, if your field is clogged, you cannot filter 100 liters. The size of water body, you not analyze a pond the same way that you analyze the Mediterranean Sea. But most important is the aim of monitoring. Are you interested in a rare species, or you are interested in a common species? And also the management cost of failure to detect low abundant species. All this information were taken into account, and now there is, we're starting from the innovation to go to an harmonization. There is several initiatives of standardization of eDNA, and all these constraints are taken into account. This is, I think, the first standardization standard that came out about environmental DNA in 2023, and is a part of the cost action DNA Aquanet. And all was taken into account, and the President slide was taken into account in this when it was built up. However, it's not finished to work on environmental DNA. Actually, there are plenty of things that need to be, we need progress for that. For example, field sampling, I think now that we really understand how it is work, what we need to sample, but some place we need really to better understand what is done, how it's the behavior of eDNA in the environment, but most of all, the reference database is very key action, because actually without reference database, the client will have bad results, and bad results means, for market point of view, also bad. No client. That's, even if there's still progress, there is a push up coming from the scientific community and also coming from the market players and the stakeholders to push forward. And now environmental DNA start to be implemented in the national strategy. Europe was already ahead of it, but now in 2003, France put environmental DNA as a tool to describe the biodiversity at national level, and also United States in 2024. So to make, taking again my title from Scientific Innovation Market Prize, making a comparison what we know, for example, artificial intelligence, the first scientific paper came out nearly in the 50s. The first commercial app started in 80s, and the global market expansion is just very new. It started in 2010, so it takes more or less 30 years for the first market action, and 60 years to mature global market. But environmental DNA was much, much faster. By the end of 2000, the first scientific study on macroorganisms, there was a rapid academic validation and the commercialization survey emerging in the 2015, and the method is recognized in the 2020s, and the market is growing. So the market adoption was much faster, in less than 10-15 years. But why this was much faster is because there was an urgent need to biodiversity understanding, readily available sequencing technologies, because without Illumina it was not possible to use environmental DNA, and strong institutional demands already present. But for me, the DNA success has been driven by the strong collaboration between academia, NGO, companies, and stakeholders, because without that, it was not possible to have this huge increase on the market. Thank you very much.
Thanks, Alice. And time for any questions?
Thank you. I'm Tiina Laamanen from Finnish Environment Institute. It was super important what you showed. I just wanted to comment. You showed that first eDNA standard which is out, and actually tomorrow my colleague Christian Meissner will tell us more that there are many more to come. So it's really nice to see that how this is developing so fast, and we as a community work on that. So thank you.
Yeah, of course, there is just the first standard that came up, but I think you're working for eDNA, so there are many much standards that will come up in the next future, and this is good because for a marketplace, for a market point of view, is something good because also clients feel more secure about the reliability of the technique.
Torbjørn?
Thank you for an interesting talk. And on the note that you just ended there, since many eDNA studies do not show the same results or maybe complementary results to traditional monitoring, and have the challenge of detecting DNA that can be transported over long distances, for instance, do you sense some kind of skepticism towards the method in the market, or are they all kind of aware of the limitations and the benefits that this method gives?
Not all the clients are aware. Actually, most of the clients, they don't know what is environmental DNA. They heard about environmental DNA, they want to use it. So it's really a role of academics and also the company to explain the limits of the method. If there is, for example, travel distance of environmental DNA through the river, that is not very, can go for several kilometers, but if you study, for example, soil, just a few meters around you. So there is a need to train the people, the end user, to use it, because behind the word environmental DNA is something like, say, genetics. So that's an entire word. So we really need to train and to people understand. And for example, we as a private company, we spend time to explain to our client what the type of environmental DNA we did, because other competitors do environmental DNA but in a different way. They have different results because they respond to different questions. So that's very important, and people are not aware yet.
Thanks very much, Alice. And so we've just had a set of talks on kind of R&D applications, and now we're kind of moving in the final talk, and Tiina is going to speak to us about how we kind of go from sets of R&D activities through to a national strategy and plan.
Okay, so hello everyone. My name is Tiina Laamanen and I'm coming from Finnish Environment Institute. And today I will tell you a little bit about our national roadmap, how we can implement eDNA. So, something about 3 years ago, we published our national eDNA roadmap. And why we did that was that we really saw that we really need to see, like a national level, that what is the state of the art with these new developing monitoring methods. And we were aiming to identify the main possibilities and also challenges and development needs. And we were aiming to formulate that kind of like a really concrete medium and short-term aims. And a really concrete action plan for the next 4 years. And as you can see, there are a really long list of experts, so it was really nice, like a national level collaboration, and we really saw quite fast when we started this project that we need that kind of like a cross-organizational interaction in practice. Even though Finland is quite a small country, but still we have quite many national level institutes, and all of those are doing their like own stuff, and as we all know, we are quite excited about eDNA and atom molecular monitoring methods, but it can cause that if there ain't that kind of like discussions ongoing, we are developing our own approaches. So we saw that it was super important to have that kind of like a national level understanding that who is doing what and why. And also, of course, as we all know, international coordination and collaboration, they are super important. So, as a part of this national eDNA roadmap, we made a survey. It was really nice effort. We got 171 response, and light blue you can see the national level answers, and orange are the international ones. And we really saw that as a community, we see that limiting factors at the moment are lack of funding, expertise, and method standards. And please, if you are willing to see more in detail, it's all in that report. But it was like a quite nice starting point for us to see that, okay, we are like on a right track, that we really need to develop those standards, and we really need that kind of like a national level and international level training events, and just to give our experts more understanding that what is the current situation and what we need to do as a community. So, as a part of that national eDNA roadmap, we made that kind of vision where we see that internationally standardized molecular methods, they really can act as a cornerstone of our routine monitoring. And as you can see, there are quite many quite concrete steps, which some of those we have already took. So one of those was that 22, this national eDNA roadmap was published. And another one, which was really important, was that 22, our Finnish Ministry of the Environment, they launched that kind of biomon funding program. And in that program, there were several eDNA-themed development projects running from 2022 to 24. So like a national scale, it was super important that we had that kind of like a funding available. And for example, macroinvertebrates, phytoplankton, zooplankton, diatoms, there were a specific group which were then developed, and for example, reference library for those were development during those national level projects. Then 23, the standardization took a quite nice steps further. So my colleague Christian Meissner will tell you more tomorrow about that ongoing standardization work, but I can already mention that there is this ISO group, eDNA, DNA, and RNA methods, and that standard. So the last presentation was the first one. It was first like a CEN, like European level, and then they made it ISO standard, international level standard, and there are many more to come. There is this kind of international eDNA standardization task force. It was launched 23, and there are international experts all across the globe working hard to get those standards in the ISO. Then 24, we published that kind of nature data portal, and the idea is that it's like a national portal where everyone can easily go, and if they are willing to know more about, for example, eDNA methods, what they are and how to use them, and what kind of like a commercial service providers there are. So that's the place where you can easily find all that information written in Finnish, because those, like, of course, national level experts, they are willing to hear that information in like Finnish. So that's what we did. And we also started planning like national eDNA data management. As we all know, it's super important that eDNA data is fair. It's really important to make that kind of like national and international level data management plans, where we beforehand plan that where and how to publish that data. So that's something that we started last year. So during this year, that international standardization of these methods has been going further. And one really nice thing was that Euromed was establishing that kind of eDNA task group. I'm leading that one, and there are many like national scale meteorological institutions and many like national level experts all across Europe, and we are aiming to think and solve that. How about the quality issues? How as a community we can work with those? So that's something that is ongoing at the moment. And also earlier this year, Ecostat eDNA task group was started, and it's really concrete. It's aiming to work on how we can implement eDNA and DNA meta-barcoding based analysis to Water Framework Directive in the future. So, and when we go a little bit further towards 27-30, we really see that these all new molecular monitoring methods, they can support our monitoring, and we really see that together as a community we can build that kind of framework where we can combine molecular data, remote sensing, modeling, and so on, and really see the bigger picture of what is happening in our nature. So just a quick mention that as a part of that eDNA roadmap project, we also started this kind of work where we were looking all of that recently published scientific papers to understand that what is the technological readiness level of these methods, how far away we are at the moment. So we looked at over 600 research papers, and we saw that actually, especially with fresh waters, we are quite close already. Like there are many that kind of organisms groups that you can actually already full scale implement these molecular methods into monitoring programs. So it's really nice to see. But then when we look those papers, that what kind of bottlenecks there are currently, many papers they were saying that, for example, reference libraries and lack of standards, they are the key barriers at the moment. So just to mention some ongoing actions, what we are doing at the moment in Finland. So as I already mentioned, those standardization efforts, there is this international eDNA standardization task force, and those draft documents are then given to ISO and CEN. Then we are doing a lot of different kind of international collaboration with GeoBon, IBOR, GBIF, and so on. So it's really nice to nice to be in that kind of collaboration efforts and workshops and so on, because we really see that Finland is so small country that we cannot do it by ourselves. We really need to go like a international collaborators and work as like a community. And we have been extremely lucky. Past few years there has been quite much funding available, and at the moment in Finnish Environment Institute we are participating many international development projects funded, for example, by Research Council of Finland, Horizon Europe, Biodiversity, European Research Council, and so on. So it has been really nice to see that we are building that kind of like a eDNA project family, as we call it, and it's really nice that all those efforts, what we are doing inside of our institute and close collaboration with our international collaborators, they are like sharing the same aim, that how we can develop these methods and how we can implement these into our environmental monitoring programs. And molecular monitoring methods are, in Finland, they are part of our national environmental monitoring strategy working group called New Methods. And this one is led by my colleague Jenni Attila. And they are also part of our national coordination group for nature information working group called New Methods for Producing and Utilizing Nature Information, led by my colleague Veera Norros. And they are also part of European Meteorology Network Pollution Monitoring Task Group eDNA, which I'm leading. So just to give you some examples that what kind of projects they are currently running. So for example, our colleague Florian Leese from University of Duisburg Essen is leading a Biodiversity Plus project called DNA Aqua Image. And there we are using diatoms and macroinvertebrates and DNA-based methods and image-based methods, and really aiming to implement these methods to Water Framework Directive. Then many of us are participating eDNA Aqua Plan. It is Horizon EU-funded project, and there are 18 partners all across the Europe, and there we are aiming to make that kind of digital landscape of what is happening now, how we can make the eDNA data fair as possible, what kind of data repositories there are currently, and how to make it work as a future. So that's really nice ongoing project at the moment. Then we have Obsession, led by my colleague Petteri Vihervaara. There are many partners all across the Europe, and there we are aiming to implement eDNA and remote sensing, how we can use these promising methods together to see the bigger picture, what is happening. Of course, Obsession is doing a lot of different things, but this is one part of that. And then in a national scale, we have this Biodiversity Life project. It's with a 50 million euros funding. It's Finland's largest biodiversity themed project ever. So it's really nice that we got that one. And there we are doing like a testing and piloting that how we can use eDNA for species composition and state of habitats. And beginning of next year, there will be new national 4 million euro project where we are aiming to solve that how and where to store the data in a national level and how to link it into international databases. So that's something that we are just about to start. So, as a summary, where we are now, as we all know, work is ongoing, there are standards are development, and there are many proof of concepts. And it's so super important to remember that international coordination is crucial for establishing common standards and ensuring that it's reliable and comparable what we are doing. So that's why we really need that kind of like international level coordination and events just like this, which we are attending at the moment, just to be sure that we are on the same page. And for that, I would really like to thank you and thank my colleagues, Christian Meissner, Veera Norros, and Petteri Vihervaara. Thanks.
Thanks, Tiina. And I mean, I think this this roadmap is a great model that could be used widely in many other countries. Any questions for Tiina?
Thank you for the nice talk. You talked about a lot of aspects that have to come together in order to standardize methodology and to make data accessible for everybody. So I was wondering, what do you see based on your experience thus far as the main constraints on finalizing a shared standard within Europe?
So like in European level especially, how to standardize, correct? Or?
And yeah, I mean, like, what are the main hurdles? Because like, there's a lot of things that need to be standardized, but some of these things are like, well, we can just pick a standard together and it's okay. But where does it get difficult?
Actually, past few years, it hasn't been that difficult. I guess there are some experts who can share that it's really nice that how experts in Europe and all across the globe, they share the same idea that we really need these ones. We cannot, because it's something that if we really want to implement these methods into full scale, it's like the first step to take. So that's, in that sense, it has been actually quite easy to get that kind of like common understanding, and it's really nice mechanism how Florian Leese, Meerat Hajibabaei, Christian Meissner and others, they built that international eDNA standardization task force, which is open to everyone. And inside of that international eDNA standardization task force, there has been so many like draft standards. And once that draft is then given to like European level, they can be given to CEN or international level to ISO. It's so much easier once it's not like a blank page where to start. There is already that draft, and then you can start to discuss on that. So it's really nice idea how they build it.
Okay, just to cut to the chase, when will it be done then?
So I think the question was, when will all the standards be in place for everything? When will all the standards be in place?
Soon. I guess because it's like the first step towards full-scale implementation. And for example, Water Framework Directive, there will be already some mentions in next stage, which starts in 27. So we are kind of hurry. So really soon, sooner than later.
Alistair's got the microphone there.
Yeah, it's on. I'm Alistair Duckett. I work in Scotland for the Scottish Environment Protection Agency. Thanks for a fantastic talk. I'm really impressed with all the development that you've been doing the last 4 years, I guess. I think my question is really about the roadmap and how much that specifically was influential in allowing you to take these steps, really.
Yes, it was really important step, and it was, I didn't remember to mention, but it was actually funded by our Ministry of Environment. So in that sense, it was like that kind of like a starting point for dialogue, that where we are now and what we need to do in the future. And I don't know, is it because we are so small country, but we have really close collaboration and connection between our ministries and our like research institutions, and it has been so like fruitful discussion ongoing. But it's so much easier to go to those discussions once you have that kind of like strategy. So in that sense, I really like that it was like a good idea to build this way.
Yeah, thank you. I'd like to find you later and speak to you more about it.
Thanks.
Another question?
Just a small question to add about the roadmap. So you're coming from the Environmental Institute, but did you involve public health companies, I don't know, agriculture maybe?
Yes, we had a national level workshops, if I remember right, 4 or 5 of those. And actually we have that kind of like a national eDNA network, and in that network we have over 130 experts. So we are really aiming to invite everyone, and those workshops and those meetings, they have been really fruitful. So that's our aim, that we can, as a like a national level, everyone are welcome.
One last question specifically. Mike, just behind you.
Thank you. Yes, that's actually a more specific question, just about the interplay between eDNA and DNA-based barcoding. And your title for the roadmap is, it sort of maybe covers both. I just wondered more specifically, and is there any sense that when you're talking about standards, I'm interested to know whether the question about lethal sampling of biodiversity from communities is part of that consideration, and whether eDNA is considered as a way of not requiring lethal sampling versus potentially DNA barcoding more directly from samples.
Yes, it's really good that you put it out. So on purpose, we use that term environmental DNA and other molecular monitoring methods. So that means that it's eDNA and it's also DNA metabarcoding. And in our report, we also describe all the new approaches, for example, RNA-based, qPCR, and so on. So it was impossible to make that kind of because we have to look in the future that what can be within 5 or 10 years. So that's why we created that kind of term, eDNA and other molecular monitoring methods. But you are correct, it's always good to be as specific as possible that what you mean when you are speaking, for example, eDNA, does it cover everything, or how to use that term?
So thank you very much, Tiina. So we're just moving now into the last kind of 15, 20 minutes or so for general discussion. And just as a preface to that, it's kind of been a real privilege to listen to the talks in this session. It was a great treat from our perspective to invite people from a range of different projects, different initiatives, and hear this fascinating range of applications, some from barcodes, some from genomes, moving on that journey from R&D around applications through to practical deployment, this importance of standards, this importance of reference libraries running through many of the talks, and then seeing how we, right at the end, translate that through to business, practical commercial operations, and national roadmaps. So thank you for all of the speakers in the session. I find that kind of extremely stimulating. So now there's a chance to ask questions. You can target questions to individual speakers as a follow-up. You can ask more general points, and we'll wave the microphone around in the direction of the speakers and see who bites. But also, I'm conscious that in the audience we've got people who have got their own kind of exciting projects that weren't featured in the talks today. So if there's just something you're working on or something relevant you wish to say, feel free just to wave your hand up and tell us about your work. So with that, I'll throw it open to the floor, and Pedro had his hand up first.
Well, thank you, Pete, for giving me the opportunity to speak in first place. And it's a general question. So we've heard of monitoring at different stages of development, from pilot studies to national roadmaps. My question now is how to do it at the European scale, because that requires harmonized methods, governance, some kind of institution coordinating the monitoring, because even the standards are not enough, because the standards are very flexible. Within the standards, there's a lot that can be done, and then the data is not comparable from one country to another. And so there are two things in monitoring that needs to be accomplished. First is comparability in space, and the second is comparability in time. And how can this be scaled up from the experience that we already have? And just getting back a little, I've just completed a review of gaps in monitoring in Europe, and even with conventional methods, there is a huge difficulty of integrating data across Europe. Even with conventional methods, even with bird counts and things like that. So how can we leverage this? How can we go beyond standards and have a governance structure, a coordination structure that makes it possible to do similar things across Europe and have comparable data across Europe?
So
it's a 1 million euros question or 1 billion euro question.
So it's great that Pedro started with a nice, simple, straightforward question. And actually, I'm delighted that it's going to get a simple answer from Tiina, but just also to flag that much of tomorrow morning is going to be focusing on exactly this topic. So I think we get some quick reflections now, and just in case we don't fully answer your question, we'll come back to it tomorrow. But Tiina.
Yes, let's do that. But there are some key things which are like easy to work on as a community, as we all can, what we all can do. So I would start that as a Europe, there must be, for example, Water Framework Directive is something that there in the future, it must be stated that where and how you collect and what you collect and how you analyze that. And of course, before going full scale, we first need some kind of like a piloting. And at least for my understanding, EcoStat working group, they are aiming to start that kind of pilot next year already. So we are already moving towards that direction. And there are many things to solve, for example, that how and where to sample and how to analyze those samples and where to store that data. But there I see also big possibilities that if we as a community can build that kind of data storing approaches, that they are then open to everyone, then it's actually better than an existing one. For example, European Environment Agency, they are saying that there is so much that kind of data that they don't actually get. There is amazing like national level efforts, but once that data is then stored to like a European scale, then it gets somehow lost in that way. So we really need as a community to do better, and we can do better. So at least I'm a bit optimistic. There are, of course, huge challenges, but there are many, many opportunities as well.
Andy?
Thank you. I agree with the last speaker. I think that this is about making sure that data are reusable and interoperable. You know, that we have common data formats, we have good metadata standards, we have open data repositories. I don't think it's realistic to think that we will have some unified program of monitoring and data collection. I think our efforts are better placed in making sure that the data are usable and reusable and that we can bring them together.
Sorry, I have 2 points. The first one is probably many of you know, but next year, the week of 4 to 8 of May, Biodiversa Plus organized the monitoring week. So I warmly recommend that this community is represented. And then to have uniformization at European level, we have the EU Biodiversity Platform subgroup on monitoring. And Biodiversa Plus is an observer, so could be a kind of ambassador. And then questions can be sent to the Knowledge Center for Biodiversity managed by GRC, like how to harmonize. So, voilà. Thank you.
I'll stand up because I'm a bit sore from sitting down for so long. Yes, Christina Hvilsom, I work at the Flanders Marine Institute. So following on about the data, okay, this is really weird. I can kind of hear myself double. The data technology to make all the data that we collect in the world available from one portal exists. It's easy peasy. You could do it tomorrow. You need, of course, the human resources, the developers, the people, to build the systems to translate the existing databases into this linked open data system, building on web technology. So you need the human resources to do it, but it's really easy peasy to do it. The direction that we have to look to get this to happen is policy. You need to say this, they need to say, this has to happen. You can't keep collecting your data and storing it in your own database, Excel spreadsheet, and not available, not findable. You have to make it happen. So that's the bottleneck. It's the requirement from policy and funders. It needs to be made a priority to these organizations that are collecting and sharing this data.
Yeah, I mean, I completely agree with that, and I'm just looking at Paul here to you know, for his reflection on the journey that you've seen in the barcoding world of where there are private records in the database, but although many of those private records remain private, they're just kind of an increasingly small part of the global database as the culture has shifted and the early release of data has become more commonplace, just because we as a community of scientists have moved from a kind of a Hobbit-like my precious approach to a Gollum-like my precious approach to data, and more of an open sharing approach. And you must have seen that operating at scale through the BOL database.
Yeah, Pete, you're right. But I mean, one of the surprising things is there's still a community of people that feel it's absolutely perfect to keep data locked up for 10 and 15 years. So ultimately it comes down to the funders, I think, that help. I mean, a culture change is happening. I mean, when I think about the kind of work that's done by the private sector, I think you've alluded to this, so much of it never moves away from the gray literature and out into the public sphere. There's massive amounts of money being spent on biological monitoring around the planet with no good outcome because the data is private. So I mean, I think it would be phenomenal if that could be one of the efforts within Europe, is if you're an industry operating under a regulatory framework, that data must move into a public database within a very short period of time. Yes, you may need time to react as an industry, but it's not good enough to put it in a gray literature repository. So I think it's the academic sector's on side. I think the public sector probably hasn't been as on side. So, but you're right, Pete, the change is happening. Open science is here.
And the perspective from kind of ERGA EBP, I'm just wondering whether Mark or Camilla wanted to comment there. Mark, you just wave an arm.
Yeah, from the EBP's point of view, a genome isn't sequenced unless it's publicly available in INSDC, the collaboration between the European Nucleotide Archive, GenBank, and the DNA database of Japan. That will change in the future as INSDC grows, but a genome has not been sequenced unless it's in INSDC, according to the EBP. And that's it's a very careful line to walk with whole genome data, because there is this issue of digital sequence information, ownership, and use, and making sure that benefits flow back to the nation states and the communities from which those specimens were sampled. But the EBP is, if you like, aggressively open.
Yeah, I would also like to respond. I fully agree as a commercial company that I think there's a lot to be gained from sharing data, because we collect a lot of eDNA data every year, and we're basically hardly doing anything with it, while it could be very valuable to other parties to do ecological analysis. So I think there's a big opportunity to involve industry in this effort, but also to think of mechanisms that can actually help us do this, because for now we can contribute our data, but it takes time, and this time is not paid for by our clients. So we have to think of either like a legal mechanism or some incentives that this is also, I guess, beneficial to us. And adding to this, I had another point to make, that like we are as an industry in the Netherlands already working as well on standardization within our ecology consultancy companies. But one of the things that we are very much struggling with in the standardization is the balance between development, openness to development and innovation on one hand, and consolidating standards on the other, because these are kind of conflicting. And I think that is a very topic that is complicating for us. I don't know how you other people view that.
Alice,
Yeah,
did you?
sorry.
Go
Do it
on.
later.
Right, Alice, just on this point, and then Camilla, if that's all right. So, Tiina, can you just pass Alice the
Just to finish the comment about the private company and the data, depends also on the government, because, for example, in France, they start a working group about every biodiversity data, even done by the eDNA, should be fair and give it so even the things that a private company will end up, the final client will give a biodiversity assessment, and this biodiversity assessment should be fair and done, and so put on the database with all the information how this data was produced. So things are moving forward, even from a company point of view. So we will need to take this to account and move forward for this.
Camilla?
Yeah, well, just complimenting, because I agree completely. I think from what Mark said, from the scientific point of view, I mean, we are even obliged to put everything that we do in open access, otherwise we don't publish nowadays. No? So I think this is a culture that was imposed on us, and luckily it was imposed on us, so we were very used and already expect that everything is going to be open out there. And I'm very happy to hear this from the industry, because it does worry me that, I mean, we are all trying here to develop the methods so that they can be used outside of the academy, that they can be used for the monitoring by governments, by other types of actors. So this is going to be a very important point, that we work together and we make it sure that this data is not only available, but is available in the most harmonized and accessible, well, fair way possible in all these spheres, also coming from governments, focal points, report points, and so on and so forth. So, yeah.
Perfect. And two last questions, because just my line of sight was Paul and Ember there, and then we'll wrap up.
I just wanted to pick up Christine with the results that you reported, monitoring invasive species, these phenomenal numbers of organisms that are coming in to Norway. I mean, at what point do we say enough is enough? I mean, monitoring is not going to solve the problem of species invasions. What you've shown is it's an unbelievably leaky sieve, and you can watch carefully some of the places some of the time, but it's going to get in unless we change our practices. And I look at the externalization of these costs. In North America, we have been introducing wooden pallets from Asia, and with them, every now and again, come in beetles that eradicate all of the ash trees in eastern North America at costs of multi-billions of dollars. Australia has had the fire ant introduced on recurrent things, and now they've lost control of it. Maybe we would have stopped that with monitoring, but most of these, the ship ballast problem, billions of dollars costs externalized. We as biodiversity scientists have shown there's a problem. At what point do we step up and say no more wooden pallets, plastic pallets, no more ballast water being introduced? I mean, we have the information. We know invasions are happening. They're really expensive for society. They don't cost industry. But I think, how do we move from those observational data to policy change?
Another nice and easy question for you.
Well, I can tell you one thing for sure is that it's very frustrating to every year detect all these species and produce all this knowledge, and then nothing happens. Yes, you could import. Yeah, you could import plants without soil as well. They could be growing in in another medium. So there are many opportunities, but they all cost money for the companies importing them.
I would love to see as a start that in our, at least in the public sector, that procurement frameworks feature a biosecurity element in the public tendering process. And that something as simple as that would be a start point, because you see lots of public projects which are, you know, issuing tenders for the delivery of activities that then pose a risk to the public purse. I mean, it's a really unpleasant cycle, and that would be one place where you could put a first block in it.
Well, actually, it builds perfectly on Paul's question and this conversation, and that's, I mean, there's the immediate reaction that I have to the idea, let's ban pallets, or let's ban, you know, plants that have soil attached to them, is try to get a politician who is the boss of the policymaker to agree to that. Because every single homeowner in that country will say, no, 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 but I want to have a garden with plants, not with seedlings in it. Right? There's a reason that we import those, is because we have an economy that's built on that. So the fundamental, you know, bad guy is globalization, and ultimately these are, you know, massive political choices that are playing out in real time now. But this goes to this, and I was really struck by Christina's mention of these genetic diversity indicators, because what I think we see there, so the example was basically, we have the tools to monitor at the DNA level. The Strategic Plan for Biodiversity for the next 30 years chose its headline indicator to not be based on those methods. That's what she saw. And actually, Belma and I, because we were both in the ad hoc technical expert group for the monitoring framework, were in the room when that was decided. And I remember sitting there thinking, this isn't a good idea, but I didn't have the power amongst consensus to influence that decision. And the powers that be, the people that have a deeper seat at the policymaking table, are still doing it the old-fashioned ways. And so the question is sort of multifold. Are DNA-based, are omics-based methods now at the point of maturity that we realistically think they should be part of the policy framework? And will Europe take a leadership role in that? And are we prepared to invest at the global level to make that happen so that we don't just go it alone? And then are we ready for the political ramifications that come from that, like no more pallets, right? Like if you can monitor at a very high level, you know, this is kind of like precision-based medicine. Like if you can monitor every single disease you might ever get, do you really want to know? Are you really ready to handle the medical choices that come with that? Are you ready to handle the political choices that come with realizing how bad we're screwing up biodiversity? So I think there's kind of layers that need to be addressed. I'm not expecting an answer, but I think that it would be really helpful if this community can say, yes, we're ready. These are the things that are ready to now put into policy practice. And this is where we'd start. You've had some excellent examples here today, but the question is, how ripe is it? How mature are we?
So I think we'll leave that as the question hanging to go into the drinks reception that you all deserve for such a productive afternoon. Now, the drinks reception in some ways is just a normal drinks reception. There will be some drinks. However, there's been such a kind of fascinating set of conversations here, and I know people have come a long way to get together. We just want to help structure the continuation of the discussions, not in a forced way, right? You can just have a drink and chat. But once we're in there and settled, what we're going to do is kind of identify 3 areas. One of those areas is going to be where there will be a place to continue some conversations around how eyeball and EBP best work together. So this kind of collaboration among the genome sequencing and the barcoding communities. Another area will be thinking about applications, you know, questions, what can we as researchers do to better help meet stakeholder needs? And then the third area is going to be around mainstreaming and a lot of the things we've just talked about. So I'm going to just stand there with a glass of wine and chat away about the application stuff. And then Rob is going to stand there with a glass of wine and chat around the EBP eyeball stuff. And Camilla and Demetrius are going to stand there and talk about the mainstreaming stuff. And we just kind of places where you might go and find like-minded folk for a brief bit of the drinks reception. So in terms of the practicalities, Gabriella will tell you where it is and what to do.
Yeah, you might have thought, well, we finally found a way to the catering room, so now we are good, but I can tell you that you are not, because the networking drink is in a completely different place. But luckily it's not far, so you just take the same way as to the catering, well, where you went to the coffee break, but instead of going right to the room, you will go to the left, up the stairs, and we are going to have the networking drinks in the mezzanine. Hopefully it's all set up so you will recognize it. So have a lot of fun, and thank you for all your attention and the great talks.